IB Bio, Topic 6, Human Physiology, covers 7 body systems: digestion, circulation, immune, gas exchange, nervous system, endocrine, and reproduction. Part 1, this movie, is the first of 2 parts on the digestion absorption system. This includes the breakdown of macromolecules that we ingest and the absorption of nutrients from the digested food into the bloodstream. The essential idea is the structure of the wall of the small intestine allows it to move, digest, and absorb food. Here is the first of 2 slides that provide an outline of all parts of Topic 6, Human Physiology, for SL and HL students. Use the outline to find the movie you need for review. This movie is focused here, and it provides a range of concepts, some review concepts, some introductory, and some fairly sophisticated related to digestion and absorption. Here is the second slide of the outline for all parts of Topic 6, Human Physiology. The primary focus of the IB syllabus is on the small intestine, seen here. As I work to provide a narrative on digestion and absorption, I will present some review and introductory concepts as we move toward a full understanding of the sophisticated detail. Heterotrophs, unlike autotrophs, cannot make their own food. Thus, as mammals, we must ingest the large macromolecules that are found in other living things. As mammals, we ingest large macromolecules. We digest those large molecules to produce the monomers that are small enough to be absorbed into the bloodstream across the layer of cells that make up the epithelial lining of the digestive tract, the epithelial lining of the small intestine in particular. And undigested material, such as cellulose, passes out in the feces. Exchanging matter with the external environment is a fundamental activity of all living things. For animals, we take in our food with the digestive system. We exchange gases using our gas exchange system, and we release nitrogen waste using our excretory system. Our bloodstream lies adjacent to each of these 3 systems. The bloodstream links cells deep within the body to each of the 3 systems that exchange materials with the environment. Here's the first IB syllabus statement to kick off this movie. Draw and label a diagram of the digestive system, including the mouth, esophagus, stomach, small intestine, large intestine, anus, liver, pancreas, and gallbladder. The diagram should show the interconnections between these structures. Here is the first of a series of images of the digestive system. Use the image that suits you to study the anatomy of the digestive system, to study the relative position of all the organs. Starting with the mouth, let's follow in chronological order. Food goes into the mouth, down the esophagus, into the stomach, and then into the small intestine. Undigested material continues to the large intestine, to the anus. The liver produces bile, and the gallbladder stores bile. The pancreas produces numerous digestive enzymes. It's located here. Both the liver and the pancreas lie to the side of the small intestine, and ducts take bile and digestive juices to the small intestine. The pancreas secretes enzymes into the lumen of the small intestine. Enzymes digest most macromolecules into monomers in the small intestine. Here's another image of the digestive system. From the mouth, food goes down the esophagus, to the stomach, to the small intestine, to the large intestine, to the anus. The liver is here, the pancreas is here, and here's the gallbladder. Now let's take a look at a diagram that provides a slightly different perspective. In this diagram, the digestive tract has been expanded, but I've not labeled the structures. Turn the movie off now or follow along with me. Mouth, esophagus, stomach, small intestine, labeled 7, large intestine, labeled 8, and the anus. Both the liver and the pancreas lie to the side of the small intestine. Ducts take bile and pancreatic juices, respectively, to the lumen of the small intestine. The mouth, the stomach, the pancreas, and the small intestine all manufacture digestive enzymes, and that's why I've labeled digestive juices in red. Bile, manufactured by the liver, stored in the gallbladder, does not have any enzymes. Bile, much like soap, emulsifies fats into small droplets. Now, the pancreas is the primary organ for secreting enzymes into the lumen of the small intestine. Enzymes digest most macromolecules into monomers in the small intestine. So let's just back up and review. Large macromolecules are put into the mouth. This is called ingestion. Ingested macromolecules are digested physically by teeth and bile and chemically by enzymes. Monomers are absorbed into the bloodstream across the epithelium of the small intestine. The monomers, once in the bloodstream, move to cells of the body to be synthesized into large molecules or used for energy. Undigested material proceeds out of the digestive tract with the feces. Cellulose remains undigested and is a typical component of our feces. Here are 2 relevant IB syllabus statements, the second of which comes back from an earlier unit of study. Enzymes digest most macromolecules in food into monomers in the small intestine. Catabolism is the breakdown of complex molecules into simpler molecules, including the hydrolysis of macromolecules into monomers. Here is a different perspective on the digestive tract. Here's our mouth, the stomach, small intestine, large intestine, anus. Enzymes are found in saliva, stomach juices, and juices from the pancreas and small intestine that operate in the small intestine. Enzymes accelerate the digestion of polymers into monomers. This is known as hydrolysis, a term that should be familiar to you. Throughout the digestive tract, pH values change. Remember that enzymes are sensitive to pH and operate best at optimal pH values. Enzymes accelerate the hydrolysis of macromolecules into monomers at specific pHs. Monomers are small enough to be absorbed into the bloodstream, and the blood leaving the digestive tract proceeds to the liver before reaching the heart. Here's another look at the digestive tract. Large macromolecules, foods, go into the mouth. Digestion begins in the mouth and continues in the stomach and in the small intestine. You can see the secretion of enzymes in the mouth, the stomach, and the small intestine. Remember that the pancreas plays a huge role in secreting enzymes into the lumen of the small intestine. Undigested material, including cellulose, goes out with the feces. Monomers are absorbed across the epithelium of the small intestine into the blood, and those nutrients move to the liver for regulation. As a note, muscles along the wall of the digestive tract move the tract to mix the food with the enzymes and to move the material along. Here's a relevant IB syllabus statement. The contraction of circular and longitudinal muscles of the small intestine mixes the food with enzymes and moves it along the gut. Students should know the circular and longitudinal muscles of the small intestine are antagonistic, as muscles can only actively contract. Here is an image of the stomach, and the diagram nicely shows circular muscles here and longitudinal muscles here. The 2 sets of muscles work in an antagonistic fashion to achieve movement of the stomach wall. As the circular muscles contract, the longitudinal muscles relax. As the longitudinal muscles contract, the circular muscles relax. This is the antagonistic nature of these two different sets of muscles. Here is a diagram of the small intestine. While we will study this diagram in more detail in part 2, you can see the circular muscles here and the longitudinal muscles here. The contraction of circular and longitudinal muscles of the small intestine serves to mix the food with enzymes and moves the food along the gut. The two sets of muscles work in an antagonistic fashion to achieve movement of the stomach wall. As the circular muscles contract, the longitudinal muscles relax, and as the longitudinal muscles contract, the circular muscles relax. I've repeatedly mentioned the role of enzymes in digestion, but in addition, it's important for you to know that different regions of the digestive tract have different pH levels. I briefly mentioned this in an earlier slide. So that brings us to these two IB syllabus statements that should feel like a review. Describe the effect of pH on the rate of enzyme activity. Sketch a graph showing the effects of pH on the activity of enzymes. In this slide, we have enzyme activity on the y-axis and pH on the x-axis. Each curve represents a different enzyme. Enzyme A, enzyme B. Enzyme A has its maximum rate at pH 2, while enzyme B has its maximum rate at pH 8. For either enzyme, A or B, any pH away from optimum results in the denaturation of the enzyme, thus less activity. Enzyme B would be an enzyme found in the mouth or small intestine, delivered to the small intestine by the pancreas. Saliva in the mouth and juices in the small intestine have a high pH. Enzyme A would be an enzyme found in the stomach. The stomach has a pH of 2. A protein digesting enzyme works optimally in the stomach around pH 2. A changing pH regime through the different sections of the digestive tract, along with different enzymes, ensures that all of the possible macromolecules that we ingest get digested. This slide should look familiar to you. Enzymes digest polysaccharides into monosaccharides in a process known as hydrolysis. Enzymes digest lipids into glycerol and fatty acids in a process known as hydrolysis. Enzymes digest nucleic acids into nucleotides in a process known as hydrolysis. And enzymes digest polypeptides, proteins, into amino acids in a process known as hydrolysis. Here's the relevant IB syllabus statement. Starch, glycogen, lipids, proteins, and nucleic acids are digested into monomers. Cellulose remains undigested. You should remember the beta-glucose structure of cellulose. Every other glucose molecule enters the chain with reverse orientation. Mammals do not have the enzyme for cellulose. Thus, cellulose remains undigested in the gut of the mammal and passes out with the feces. So as this movie is meant to be an introduction to digestion, let's start back at the beginning just for a minute. Apologies for the redundancy. Feeding starts with the mouth, ingestion of polymers, large molecules that require digestion to produce monomers that can be absorbed into the bloodstream. Teeth play a role in digestion as they physically break food into smaller pieces. Bile plays the same role for lipids. Teeth and bile physically digest food. Enzymes chemically digest food. Mammals have teeth to grab and hold prey, but also teeth are there to break food into smaller pieces. Smaller pieces of food have a higher surface area to volume ratio. Thus, with greater surface per volume, enzymes are better able to bind and chemically digest the macromolecules. Here is 1 cubic centimeter of food as a single piece, and here is 1 cubic centimeter of food as 8 pieces. The total surface area of the 8 pieces is twice the surface area of the 1 piece. Enzymes start to break down at the surface, so the higher the surface area to volume ratio of the food, the faster digestion occurs. In addition to the physical digestion that occurs by teeth and bile, glands secrete enzymes for chemical digestion. These glands are known as exocrine glands, and they include the salivary glands of the mouth, stomach, pancreas, and small intestine. But the focus of the IB syllabus is the pancreas secretes enzymes into the lumen of the small intestine. Saliva has salivary amylase. Amylase is capable of digesting amylose, or starch. You can see the salivary glands that surround the mouth in this image. Glands that make enzymes are called exocrine glands because the enzymes move in ducts, small tubes, into the digestive tract, in this case, into the mouth. As we swallow food, the food is moved into the esophagus, heading toward the stomach. The food is pushed away from the trachea, this is the trachea, by the epiglottis that keeps that food out of our gas exchange system and keeps that food moving in the esophagus toward the stomach. The esophagus is here, more to the dorsal or back side of the body, relative to the trachea, which is here, more to the ventral or the lower side of the body. In cross-section, you can see the relative positions of the esophagus and the trachea. The trachea is held open by cartilage rings. This is important due to the need for continuous exchange of gases from the atmosphere. Again, here we can see the relative position of the esophagus and the trachea. The esophagus is dorsal to the back side of the trachea, and the trachea is held open by cartilage rings to ensure unobstructed flow of air into the lungs at all times. Feeding is more periodic, thus the esophagus can collapse. And food is moved along the digestive tract by the contraction of circular and longitudinal muscles. In this image on the right, food is moved along the esophagus to the stomach. The active movement of antagonistic muscles, the circular and longitudinal muscles, the active movement of those muscles along the digestive tract is called peristalsis. In this cross-section of the esophagus, you can see the lumen of the digestive tract, and you can see the muscle layer. The contraction of circular and longitudinal muscles of the small intestine mixes the food with enzymes and moves it along the gut in a process known as peristalsis. The circular and longitudinal muscles of the small intestine are antagonistic, as muscles can only actively contract. Here's the relevant IB syllabus statement. The contraction of circular and longitudinal muscle of the small intestine mixes the food with enzymes and moves it along the gut. In addition to the muscular wall of the stomach, there are glands that secrete enzymes for protein digestion. The pH in the stomach is low, optimum for the enzymes that function in the stomach. Enzymes are secreted by glands in the stomach, located here among the folds. These folds are called gastric pits. And from the stomach, macromolecules are moved into the small intestine, where numerous enzymes from the pancreas and the small intestine itself enter the lumen of the small intestine. Enzymes digest most macromolecules into monomers in the small intestine, and the monomers are absorbed into the bloodstream, and the blood moves those monomers to the liver for regulation. Macromolecules leave the stomach for the small intestine. The liver delivers bile. Bile is stored in the gallbladder. Bile physically digests fats into small droplets. The pancreas secretes enzyme into a duct that takes those enzymes to the small intestine. Enzymes digest most macromolecules into monomers in the small intestine. And then on to the next part of the series, bringing us to the end of IB Bio Human Physiology Part 1, Digestion Absorption A.